Okay, welcome back. So
Right.
we are talking about the functions of carbohydrates, and we are talking about them having a protein-saving action or sparing proteins. So I said that the presence of adequate glucose basically spares the breakdown of proteins from being used to make glucose that is needed by the body. So they have a protein-sparing function. The other function is that they also assist in lipid metabolism. So they assist in lipid metabolism. Basically, one of the effects is that they have a fat sparing effect. Remember, we had a protein sparing effect. We also have a fat sparing effects that carbohydrates have. So, for example, if blood glucose levels rise or elevate, we find that the use of lipids as an energy source is inhibited. So when you look at lipids can also be used as a source of energy. However, if there are high levels of glucose in the blood, lipids are spared from being energy sources. And then carbohydrates also prevent the development of what is called ketosis, or having elevated ketones in the body, having elevated ketones in the body. So we find that ketone bodies really become an alternative source of energy when glucose supply is insufficient. For example, if someone is fasting. So when you look at the ketone bodies, they are acidic in nature. So if there are high levels in the blood, they're going to cause the blood to become too acidic. So we find that this is most common in alcoholics. So people that take alcohol. So it's common in alcoholics, or in someone who is malnourished, or people with type 1 diabetes mellitus. So they are common in that. And then the other function is they play an important role in cellular recognition processes. So, for example, in the recognition between a sperm and an oocyte or an egg during fertilization, or in the return of lymphocytes in the lymph nodes, or for example, in leukocytes or white blood cell adhesion to blood vessels. So they play an important role in cellular recognition processes. So carbohydrates are important for energy production or immediate energy provision. If there is excess glucose, that excess glucose is stored as glycogen in animals or as starch in plants. So carbohydrates function as energy storage. Then we said that carbohydrates also function for building macromolecules such as nucleic acid and also lipids. And then we said that carbohydrates also function as structural support or as structural element. So we talked about cellulose, which is found in plant cell walls, and we talked of chitin, which serves as a structural element in exoskeletons of arthropods. And then we also talked about carbohydrates having a protein-saving action. And we also said that carbohydrates are involved in lipid metabolism, and they are also important in cellular recognition processes. Cellular recognition processes. So we said that basically the carbohydrates are divided into three main groups. We said monosaccharides, disaccharides, and polysaccharides. So when we look at the monosaccharides, these typically contain from 3 to 7 carbon atoms. Remember, I said that the least number of carbon atoms that we find in carbohydrates is 3. So in a monosaccharide, to find that a hydroxyl group is bonded to each carbon atom except for one. So remember I said that the carbohydrate contains a lot of carbon to carbon bonds, and to that we also have numerous hydroxyl groups bonded to that carbon skeleton, and also the carbonyl group. So I find that on each carbon atom there is a hydroxyl group except for one carbon atom. That carbon is double bonded to an oxygen atom, forming a carbonyl group, the one that we talked about, which could either be an aldehyde or a ketone. So we said if the carbonyl group is at the end of the chain, the terminal carbonyl group is what is called the aldehyde. And then if that carbonyl group is at any other position, which means an intermediate position, it means that that monosaccharide is a ketone. So, and then we mentioned that the large number of polar hydroxyl groups plus the carbonyl group gives the monosaccharides hydrophilic properties, and henceforth the carbonyl groups and the hydroxyl groups are very important for the reactions that monosaccharides participate in. So when you look at the monosaccharides, the carbon atoms are actually numbered. So by conversion, so this is simply by a conversion. People just sat and decided that okay, the numbering should be in this way. So they are numbered beginning with the carbon at or nearest the carbonyl end of the open chain. So they are numbered from the more oxidized end, as we are going to see. So we have what are called trioses. So trio means 3. So these are the simplest carbohydrates that we have, the trioses. So it simply means that they have 3 carbon atoms. So these examples we have glyceraldehyde and dihydroxyacetone. Glyceraldehyde and dihydroxyacetone. And then we also have the pentose sugars. Commonly, we have ribose and deoxyribose pentose sugars. So these contain 5 carbons. And as we have already looked or alluded to, we said that these are major components of nucleic acids. And then we have hexoses. Hexoses. So hexoses are very important energy molecules. They are very important for energy. So examples of these are glucose, fructose, and galactose. So important hexose sugars or 6 carbon sugars are glucose, fructose, and galactose. So sugars that are contain 5 or more carbons are rarely found in linear chains. So they are rarely found in linear chains. So what they do is that when they are in aqueous solution, they tend to form ring structures. So that bond that is responsible for ring formation occurs only between the carbon containing the carbonyl group. So I have that carbon that contains the carbonyl group and one of the carbons with the hydroxyl group. So for the formation of the ring structures, the carbonyl group will participate. And also one of the carbons with the hydroxyl group. So the reaction occurs between the carbonyl group, very important, and one of the carbons with the hydroxyl group. That leads to the formation of ring structures or ring forms of the molecules. So especially, for example, the pentose sugars and also the hexose sugars, they are usually found in ring form. So we find that there are many different types of monosaccharides that exist because of the variability in their structures. So for example, we have aldoses, we have ketose, we have variation in the number of carbons, we have different arrangements of hydroxyl groups in space, and there are also alternative ring form. So we find that each monosaccharide will have a unique structure and function. So the monosaccharides have a variety or different aspects of their structure, which gives variability in their structures and functions. So this shows examples of monosaccharides that we have. Remember, I said that conventionally, the numbering of the carbon skeleton of a sugar starts with the carbon at or nearest the carbonyl group, at or nearest the carbonyl group. So let's take the example of glyceraldehyde in its straight chain, or in its chain form. So remember, glyceraldehyde it has an aldehyde carbonyl group. So it has a terminal carbonyl group. So that carbon bonded to the oxygen by a double covalent bond, that carbon will be the first one, it will be numbered as 1. And then the next one will be 2, the other one will be 3. So for example, another example we have there is a pentose sugar, ribose and deoxyribose in their ring form. So I'm going to look at how these ring forms are formed as we continue with the lesson. However, we have the numbering also in the ring form. So we have number 1 there, number 2, number 3, number 4, and number 5. Remember the difference between the deoxyribose sugar and the ribose sugar is on position number 2. So very important, carbon number 2. Very important, that's where the difference is. We have a hydroxyl group, OH group, on carbon number 2 in a ribose sugar, or we have a hydrogen atom on carbon 2 in a deoxyribose sugar. Then we have the hexose sugars there, examples of the hexose sugars, also are numbered as 1, 2, 3, 4, 5, and 6 in their ring form. So when we look at the trioses, the trioses. So we're going to look at the trioses and then the pentose sugar, then we'll end from there for today. So the trioses, trio, we said that it means 3. So of biological importance are 2 triose sugars. So glyceraldehyde and dihydroxyacetone. So you have C3H6O3. So they have the same, they obey the general molecular formula of 1 to 2 to 1 ratio, 1 to 2 to 1 ratio. This is a triose sugar. So remember, the glyceraldehyde is an aldehyde carbohydrate. It has an aldehyde carbonyl group. So it has a terminal carbonyl group. And then when you look at the dihydroxyacetone, it is a ketone. So it has a carbonyl group intermediate. It is not a terminal carbonyl group. When we also consider glyceraldehyde, we find that this one has 2 optical isomers. So glyceraldehyde has 2 optical isomers. So we have the L glyceraldehyde and the D glyceraldehyde. So L, remember I said that it's levo and D is dextro. So these are very important molecules, especially when you start looking at respiration or cellular respiration. So I find that when you look at a process that is called glycolysis, that is involved during cellular respiration, find that a molecule fructose 1,6 diphosphate is actually broken down into glyceraldehyde 3-phosphate and also dihydroxyacetone phosphate, which leads to the production of either lactic acid or pyruvic acid and ATP. So they are very important during cellular respiration. And then we have the pentose sugars. We have the pentose sugars. So the pentose sugars, pentose means 5. So they have 5 carbon atoms. So they have 5 carbon atoms. So they are aldehyde, right? Because they have a terminal carbonyl group. They have a terminal carbonyl group. So there are 5 carbon sugars, and they have the formula of C5H10O5. Example, in the example of a ribose sugar. So the ribose sugar has a C5H10O5, which obeys the general rule of 1:2:1 ratio. But however, when you look at the deoxyribose sugar, it deviates from the CH2ON general molecular formula because it has 5 carbon atoms, 10 hydrogen atoms, but 4 oxygen atoms. So remember, because of position 2, a ribose sugar has a carbohydroxyl group on position 2. It has an OH, while the deoxyribose sugar has simply an H on carbon number 2. So one of the molecules that does not have the general formula, does not follow the general molecular formula or empirical formula for carbohydrate is deoxyribose sugar. So it does not follow the formula of CH2ON as we have seen. So a deoxyribose sugar. So the formation of ribose furanose ring. So remember we said that the pentose sugars generally or rarely are found in chain or in straight chain forms. They are usually found in ring form in biological system. So they form rings. So how do they form rings? So we have carbon 4. Carbon 4 or combine with carbon 1. Remember we said that the carbonyl group and one of the hydroxyl groups combine to form the rings. So for the ribose sugar, it is carbon 4 combined with carbon 1. So I was saying that carbon 4 will combine with carbon 1 to form a 5-membered furanose ring for both ribose sugar and deoxyribose sugar. So the oxygen, when you look at the oxygen of the carbonyl group, when you look at the oxygen of the carbonyl group, this double bond, the oxygen attached to the double bond of the of carbon on carbon number 1. When you look at that one, it is going to combine with the hydrogen of carbon number 4. So the oxygen at the carbonyl group and the hydrogen of carbon 4 are going to combine to form an OH group. So that OH group is either above the plane. If it is above the plane, is what is called a beta. It's called beta. And if it is below the plane, it is called alpha. So if it is above the plane, it is called a beta deoxyribofuranose ring, or if it is below the plane, it is called alpha deoxyribofuranose ring. So when you look at the alpha and beta forms, they are a result of the asymmetric or chiral C1. Chiral C1, which is attached to 4 different groups of OH, the O, the C. So the OH, the H, the O, and the C. So it's a chiral center, which makes it to be asymmetrical. So this is how the furanose rings are formed in a pentose sugar, a ribose sugar, and also in a pentose sugar. So know that carbon 1 and carbon 4, the reaction takes place between carbon 1 and carbon 4 to produce that furanose ring. Produce that furanose ring.